Dementia is an umbrella term for a group of symptoms associated with brain decline, such as memory loss. There are more than 400 types of dementia, but Alzheimer's disease is the most common. Understanding the risk factors that underpin dementia raises the hope of breaking the link. A new study advances our understanding of the genetic component to Alzheimer's disease. Scientists in the study, published in the journal Alzheimer's and Dementia, identified 13 genetic variants that increase the risk of Alzheimer's disease. To identify these mutations, a team of researchers from Boston General Hospital, USA, and the Department of Neuroscience at the University of Sheffield, UK, used a genetic technique that allows reading the entire genome, called whole genome sequencing, WGS. Unlike previous studies, which have sought to find common genetic traits that predispose to developing this disease, this time the researchers focused on finding rare variants. Contrary to what one might think, these rare variants, although they specifically repeat very infrequently from one individual to another, make up approximately 77% of the 50 to 60 million genetic variants that make up each person's DNA. As such, they are often the key to understanding the origin or development of many diseases. Dementia, the food to eat in your 60s to lower your risk. Major new study finding. The researchers arrived at their conclusion by crunching data on 2,247 individuals from 605 different families, in which several members suffer or have suffered from Alzheimer's. They subsequently found 13 specific variables associated with the risk of developing the disease. This finding could then be replicated in another cohort of 1,669 unrelated individuals. Analysis of these variants: FNBP1L, CEL1L, Link00298, PRKCH, C15 or 41, C2CD3, KIF2A, APC, LHX9, NALK-NA2, Site3 and CLSTN2, revealed in most cases to be related neuroplasticity, synaptic function and neuronal development, all of which play a role in the development of Alzheimer's. Fatty liver disease, 3 signs on your skin, insight: the breakfast drink to avoid in the morning, advice: stroke, the fruit that has risk of stroke, tips: for example, in Alzheimer's patients, 4 of these genes, APC, NA2, KIF2A, and NALK, were expressed, i.e., the information contained was synthesized into a protein, primarily in brain tissue, while PRKCH expression was significantly reduced in the temporal cortex, involved in functions such as memory and language. 2 others, however, interacted with previously known Alzheimer's disease-related genes, FNBP1L and KIF2A. All these findings, in principle, could have future applications as important diagnostic tools and even as targets for gene therapies aimed at reversing the effects of the disease. However, the authors acknowledge that the scope of the findings is limited by 2 issues. The main one is the size of the discovery sample, which is relatively small compared to those used in many genome-wide association studies, GWAS, of common variants. This is due, they explain, to the limited availability of individuals meeting the required criteria, families with several cases of Alzheimer's. However, this may in part be attributed to the fact that 80% of the sample was composed of individuals of European descent, which limits the universality of the results. Other risk factors: several other risk factors are known to increase your risk of developing the condition. Age is the single most significant factor. The NHS explains, the likelihood of developing Alzheimer's disease doubles every 5 years after you reach 65. But it's not just older people who are at risk of developing Alzheimer's disease. According to the NHS, around 1 in 20 people with the condition are under 65. This is called early or young onset Alzheimer's disease, and it can affect people from around the age of 40. Other risk factors include Down's syndrome, head injuries, cardiovascular disease,